right, so in this slide we're seeing some data, spirometric data. So what this shows is lung volume, or sort of airflow into and out of your respiratory tract over time. And what you see here then are what we call different volumes and capacities. So if you'll notice here, we have these smaller lines that kind of hover within a pretty narrow range of about 500 milliliters of air or so. We call this tidal volume. Tidal volume is the small volume of air that you're actually breathing in and out during normal, quiet breathing, which is what you guys are doing right now, right? So you're just sort of sitting down, not really having to forcibly inhale or exhale. You know, just normal, quiet breathing we call tidal volume. It's only about 500 milliliters or so. It's not a whole lot. 500 milliliters, by the way, is this is a 1 liter water bottle, so it's about half of that. Is about the amount of air that you're going to be breathing in and out, you know, when normal quiet breathing, which you guys are undergoing right now. But we understand that's not our limits of different volumes and capacities. So what we're looking at here is basically data from a device that measures this change in volume across our respiratory tract over time. So we see this is tidal volume. So you have inhalation, exhalation. So that's one sort of, you know, ventilation cycle here. And then what we see here is if you can forcibly inhale to your maximal degree, what we see here, this is called inspiratory capacity. And if you look here, a normal value, this is for a male, but a normal average value is 3.6 liters. So it means that almost 4 of these water bottles you can inhale forcibly. So you go to the maximal level, almost 4 liters of this. So it's quite a bit. Right? Now, if you forcibly exhale from that peak breath all the way down to where you can't exhale anymore, we call that vital capacity. So a normal vital capacity is about 4.8 liters or so. Okay, so if you go inhale to maximal level and then exhale completely all the way till you can't exhale anymore, that's vital capacity. Okay, so 4.8, almost 5 liters, you guys. That's a lot of air. This is sort of an average, and there's sort of normal ranges for males and females. And what these are showing are just sort of as like an average or normal range for a healthy individual. Now, what you'll notice here, though, is at the base of this breath, there's some residual volume. And residual volume is that little bit of air that you can't totally exhale, because the only way you can get all of the air out of your lungs is if your entire airway collapses, right? What that would mean then is, so this is the residual volume, is the amount of air that's left behind after you fully exhale, right? So you go, and you can't exhale anymore. Obviously, I could, but I needed to talk. So, but, you know, at the base of your exhale, if you can't exhale any further, then we have some volume of air left behind our lungs. That's residual volume, about 1.2 liters. It's not a whole lot, but the reason why there's residual volume is that in order to get all of the air out of your lungs, they have to fully collapse. All of your airways would have to snap shut, leaving no air left behind in the lungs. So we know that doesn't happen because, for one, the airways are kept open by those tracheal cartilages.
So I guess I'm maybe overthinking. Vital capacity, it doesn't make sense. Because technically, like, when you're breathing in, that would be an increase in volume, but wouldn't exhaling be a decrease in volume?
Yes.
So wouldn't you be, the numbers you add, wouldn't one be positive and one be negative, so that 4,800 would be less than the 3,600? You have a negative volume.
Yeah, you can't have a negative volume. I think I know what you mean, though. These values are in reference to 0. So if you look at here on this this side of the graph here, so it goes from 0 to 6,000. So what these values are just the change in volume. But I think I understand what you're saying. Like if you go from the peak of your inspiratory reserve volume all the way down to the base of your expiratory reserve volume, then you might think that there could be a negative number to give you some directionality, right? Is that what you're saying? Yeah. Yeah. They just don't do that when they calculate these values. I know what you're saying. Like sort of like air loss versus air gain. So like air gain would be like inspiration.
Like exhalation as capacity.
I know what you mean, yeah. But think of it more as like the capacity to exhale, because not everyone has that same ability.
Okay.
Due to certain lung diseases, you might lose your ability to exhale a certain degree of air. And this can relate to a variety of factors like lung diseases affecting the tissue and compliance. But residual volume, just the air that's left behind in your lungs. You can't get it all out because otherwise that would require your entire airway to collapse. And total lung capacity is your vital capacity plus residual volume, which is about 6,000 milliliters or 6 liters in the average male. So if you think about it, it's kind of a lot of air. Now, the reason why your lungs can expand so much is, for one, they're elastic. And you might wonder, how the heck do you fit 6 liters of air inside your lungs? Well, some of it's actually compressed, right? When you inhale, some of it actually gets compressed inside your airways. Your lungs also expand quite a bit. So that expansion also could accommodate 6 liters. It's quite a bit of air. Now, the reason why we want to go over these normal values is that we can use this sort of data to figure out how healthy someone's lungs are, right? Like, let's say if their lungs were less compliant, like less stretchable, that would affect some of these volumes, right? Like if their compliance was low, it might not affect tidal volume. Because what's tidal volume?
Resting, quiet breathing.
Yeah, quiet breathing, right? You don't need a whole lot of lung expansion for quiet breathing. So that might not affect tidal volume. But what if someone had something like emphysema, where their lung tissue doesn't expand as quickly, it's more resistant, it's less compliant to stretching, right? How might emphysema affect something like your vital capacity, which is a measure of the total amount of air you can inspire and expire, or inhale and exhale, right? If your lungs couldn't expand that well. Yeah, it'd probably be decreased, right? You might find then that you actually have less air at the peak of your inspiratory reserve volume, right, than you could actually maximally intake. You might even have some difficulty with exhaling too, because if the lungs are less compliant, then they're going to be more resistant to even being compressed, not just stretched. So then you might actually see a deficit in expiratory reserve volume as well. So this actually might be a lower number, which is interesting. So when we get to pathophysiology, we'll actually look at some data like this to determine whether we're looking at a disease that is due to obstruction of the airway or restriction of airflow. And so we'll see some of these data later. So what we're going to do next, you guys, is talk about the exchange of gases around your body. So far we talked about ventilation and respiration. We talked about all those parts of your airway that aid in the process of ventilation or airflow. Then we talked about the parts of your airway that are involved with respiration or gas exchange. Now it turns out there's 2 types of respiration or gas exchange. We have external respiration and internal respiration. External respiration means the diffusion of gases between blood and atmospheric gas, like in your lungs. Internal respiration means the exchange of gases between blood and your body's tissues. But remember, this is all diffusion, so gases only move due to differences in concentration. Not pressure, but concentration gradient, right? So if someone had more oxygen inside their alveoli and less oxygen in the bloodstream, which way would oxygen flow? Into the blood, exactly. And would that be external or internal respiration?
Internal.
External. Because you're changing gases between air in your lungs and your bloodstream. And then internal respiration is diffusion of gases, between your body's tissues, like blood in your tissue or tissue and blood. So what if the concentration of carbon dioxide was higher inside of like muscle tissue, and the concentration of carbon dioxide was lower inside your bloodstream?
Into the bloodstream.
It would be internal respiration, and CO2 would diffuse into the bloodstream. Good. So that determines the direction of gas movement. So both of these processes rely on the physical properties of gases, like, how do these gases behave? It turns out carbon dioxide is way more soluble in water than oxygen. Like, you can dissolve a lot more CO2 in water than oxygen can. So it's easier to transport in that regard. Or the composition of gas inside your lungs too. Like, what are you inhaling? And that's gonna determine how these gases flow. Normally we should get rid of carbon dioxide out of our blood, right? Because typically the concentration of carbon dioxide is higher in your bloodstream and lower in the alveoli. But what if you started like breathing into a bag? And you're breathing into a bag. It's just going in and out, right? Well, what are you doing? You're mixing the same gases around, right? So over time, the concentration of carbon dioxide will rise inside that bag. And when you inhale it again, that means the concentration of carbon dioxide will be higher now. Not higher necessarily, but it will be higher than atmospheric concentration inside your lungs. So then you get less CO2 moving across your blood and lungs. And that means carbon dioxide levels will build up in your bloodstream. We'll come back to that kind of stuff later. So when we talk about the concentration of gas, we refer to something called partial pressure. Now, the thing about gases is that they are molecules that have a lot of kinetic energy. In fact, gas are molecules that move really fast, right? And these molecules are moving so fast that they start to bounce off walls of whatever's containing them. So like, for instance, if you have gas in a bottle, there's pressure in that bottle. And the pressure is all dependent on how many of those molecules and how much those molecules are bouncing around inside that vessel, right? Now, not all gases bounce around with the same degree, so they actually have different partial pressures. But this is also typically dependent on their concentration. But it's all, and when you guys take chemistry, you'll learn how it's also dependent on other physical properties of gases. But for now, we'll just talk about partial pressure as referring to concentration. So we talk about like the partial pressure of oxygen and say that oxygen has like, a certain mmHg or pressure to it. We'll just think about that as being a concentration of oxygen. Although those aren't necessarily always the same, it typically kind of goes in that particular tendency, if that makes sense. So partial pressure is the pressure that's exerted by each gas and mixture, and it's proportional to its percentage. Now, atmospheric pressure that is standardized as 1 atm, or 760 millimeters of 760 torr, okay, which is the same as millimeters of mercury. And that's partial pressure of gas. I'm sorry, that's the pressure of gases in the atmosphere at sea level. Okay. Now, the partial pressure of gases in the atmosphere in Colorado is less. There's less pressure here. For one, there's actually less air between us and space. So because there's less air between us and space, there's a smaller column of air that's bearing down on us, so the air here is thinner. But if you're closer to sea level, there's more air between you and space. There's more air kind of being drawn downward due to gravity, and that exerts a larger pressure. So the partial pressures of gases in Colorado are different than they are at sea level. Now, what this slide is showing you guys is blood as it's moving across a capillary bed in the alveolus. So is the alveolus part of your respiratory zone or conducting zone? Respiratory zone, right? So that means where we can get gas exchange there. So if you can imagine then, if deoxygenated blood, by the way, you guys see how it's PO2? And see how the units are millimeters of mercury? Well, that's pressure. But remember, we're talking about gases, their pressure as being the same as concentration. So if we say that the concentration of oxygen is low, like 40 here, in what we call deoxygenated blood. Now, that's a misnomer because you can't ever really fully deoxygenate blood. It's never going to be zero oxygen in that blood. It just means it has less oxygen than you could. So when we say deoxygenated blood, we really mean blood with a partial pressure of oxygen around 40 millimeters of mercury. So less than you could have. Now, as that blood, deoxygenated blood from pulmonary arteries, goes across the capillaries of your alveoli, it slowly, actually pretty rapidly, sorry, take that back. It pretty rapidly oxygenates, right? So why does the blood become oxygenated here? You can see that it almost triples in concentration. We go from 40 to 104. Gas exchange, you got it. And what determines the direction of gas movement here?
From high to low.
Concentration, you got it. So because oxygen is in low concentration in the bloodstream, oxygen in the gas of your alveoli will have a tendency to want to diffuse into the blood, and as it diffuses into blood, that makes blood more and more oxygenated, right? Until we get to a maximal level. They've got this plateau here, and this plateau basically tells us, well, this is the maximum amount of oxygen our blood will be able to absorb. And that's dependent on the amount of hemoglobin you have in blood, right? Which is also dependent on the number of red blood cells and your hematocrit. So we talked about that earlier. And you can see here that your blood can fully oxygenate in the matter of like a quarter of a second. So it's pretty fast, right? That's 250 milliseconds. That's about the time of like a slow reaction time. Like if we tested you guys' reaction time, a slow reaction time would be like 250 milliseconds. Like if I asked you guys like to push a button in response to a sound. If you were really slow, that'd be 250 millisecond reaction time. But that's still pretty fast. Like you wouldn't know it was slow unless you actually measured it. But the reason why it oxygenates so quickly is the diffusion membrane is thin. Remember we said that diffusion distance is inversely proportional to rate. So that if our diffusion membrane is thin, then oxygen gets diffused into the bloodstream very quickly. If you increase that diffusion membrane, or respiratory membrane, if you increase the thickness of that membrane, it would take longer for oxygen to diffuse into blood. So that the slope of this line would be less steep. It would be more gradual. And it might take you, almost a full second to oxygenate your blood. And that could be good or bad. Depends on what you're doing. But it's just something to keep in mind. So when we say that blood is oxygenated, we mean that the partial pressure of oxygen in that blood is 104 millimeters of mercury. When it's deoxygenated, is it fully lacking oxygen? No, it just has less, right? About close to 3 times less. So what this slide is showing to you guys is basically the composition of gases in different air or tissue around your body. And it also shows external versus internal respiration. So which type of respiration involves the movement of gases between blood and body tissues? Internal. You got it. So that's the one you can see down here. This is internal respiration. And this is external respiration because it's between blood and the air of your alveoli. So if we look here, you guys, we see that inspired air, this is air from the atmosphere, has about 160 millimeters of mercury of oxygen and only about 0.3 millimeters of mercury of CO2. So carbon dioxide concentrations are fairly low in Earth's atmosphere. It's less than 1% or so. Oxygen concentrations are higher in Earth's atmosphere because there are organisms on this planet that make oxygen, right? And they actually keep the concentration of oxygen high in our atmosphere, and we're dependent on that for our survival. Now, when you inhale this air, you guys, initially it has a lot of oxygen and not so much carbon dioxide. But after you inhale this, and once that air gets to the alveoli, wait a second, the concentration of oxygen is much lower here now, right? And CO2 is in higher concentration. So what happened in the air of your alveoli? Yeah, you get some gas exchange, right? Oxygen was absorbed, which lowered the concentration of oxygen in the air of your alveoli because it was absorbed in your bloodstream. Carbon dioxide levels are higher because carbon dioxide was expelled. CO2 moved out of blood into the alveoli here. So you see the concentration of carbon dioxide increased, close to 100-fold, which is pretty cool. And so the reason why O2 levels are lower is we absorbed oxygen in our blood. And the reason why CO2 levels are higher is that we expelled that CO2 into our alveoli. What would happen to this air next? Yeah, you breathe it out, right? You would exhale it. And what you exhale then would have less oxygen and more carbon dioxide. And so we're putting carbon dioxide back into the atmosphere when we breathe. So it's kind of interesting. And then, plants and other microorganisms can use that for their own metabolism. Now, what happens then, you guys, is that oxygenated blood here, which has about 100 millimeters of mercury or so, eventually goes back to your heart through the pulmonary veins, back to the left side of the heart, where the left side of the heart pumps it out to the systemic circuit, which goes to your body's tissues. Once that oxygenated blood gets to capillaries of your systemic circuit, oxygen moves from blood into tissue. What determines the direction of this gas movement? Concentration gradient, right? So what's keeping the concentration of oxygen low in the tissue? Cellular respiration, you're right. The cells are using that oxygen. And as they use it, oxygen concentrations are staying low. And that's why oxygen will move from blood into tissue because it's in higher concentration in the blood. For one, because we picked up oxygen in our lungs. And two, because oxygen's in lower concentration in our tissues because the cells are using it. And as they use it, they convert that to water, right? But they also make CO2 as a byproduct, as a waste product, right? So carbon dioxide is expelled or sort of, released by cells that are undergoing cellular respiration. And what determines the direction of CO2 movement here? Yeah, concentration, right? So where's the concentration of CO2 higher then? Yeah, exactly. So CO2 concentrations are higher in the tissue versus our blood, which is what we can see here. If you guys look here, CO2 levels are 40 millimeters of mercury in this blood, whereas CO2 levels here are 45 millimeters of mercury. So there's higher concentration of carbon dioxide in the tissue than the blood, which means that CO2 will flow from tissue into blood. Now we call this now deoxygenated blood. However, did it lose all of its oxygen? No, we still see that there's about 40 millimeters of mercury or so left of oxygen in the bloodstream, right? We still call it deoxygenated blood. And one of the reasons why the show is blue, you guys, is that veins near the surface of your skin look kind of blue. But the blood isn't really blue. It's just a different shade of red. But because the veins are close to the surface of your skin and only the kind of light that passes through skin is more in the blue spectrum, veins then appear to look blue, which is why we show them as blue on these anatomy drawings. But to be honest, it's just different shades of red. You don't actually have blue blood. That's not a thing. There are animals that have blue blood, but not us. So that means this deoxygenated blood that's leaving the tissues comes back to the heart, to the right side of the heart, where the right side of the heart pumps it back out to the lungs, and that's taking this deoxygenated blood through the pulmonary arteries to the capillaries of your alveoli, where it can participate again in external respiration. All right. So what you guys want to know from this slide then are the factors that determine the direction of gas movement, right? So you should expect that under normal circumstances, the reason why you can absorb oxygen from alveoli in the blood is because concentration gradient. There's a gradient there, right? So why is O2 concentration higher in our alveoli than in the bloodstream? Yeah. Well, because it's higher in the atmosphere, and you inhale that air, right? And why is it low in the blood? Yeah, your cells were using it, which kept the concentration low. So that means that O2 is getting absorbed. Good. We'll talk about where it goes when it gets absorbed. And then why is CO2 in higher concentration in blood versus the air of your alveoli?
Because your tissues have taken the oxygen from your blood. Right?
That's partly why. But the cells also make CO2. Your body cells make carbon dioxide. It's a waste product. You got it. In fact, CO2, when it dissolves in the water of your body, forms an acid called carbonic acid. So by expelling CO2, then you're also getting rid of acid from your blood. So the reason why carbon dioxide gets removed from your bloodstream is that CO2 levels are higher in your blood than in the atmosphere. But what if you found yourself in a weird environment where there was like a significant amount of carbon dioxide in the air that you're breathing?
Oh, it would stay in your body.
It might stay in your body, or you might absorb some CO2 into your blood, right? So there's a weird CSI episode some years back, you guys. I don't know, maybe I was on the show, but the premise was that a college roommate killed their roommate, the other roommate, using dry ice, because that's carbon dioxide ice.
Yeah.
And then they let it evaporate. You get the carbon dioxide gas, and apparently it was enough carbon dioxide gas to suffocate that person. And the way they caught him was that there was a hole that was drilled in the wall or something like that. It's totally fictional,
Right.
but it could make sense with respect to knowing that if you're inhaling a lot of carbon dioxide, if the concentration of carbon dioxide is higher in your inhaled air than your bloodstream, then you will absorb that CO2. So the only thing that determines the direction of gas movement is concentration gradient. So keep that in mind. Oh yeah, well that's a different process though. So, but you're right. That does change concentration gradients. So how is oxygen transported in blood? Well, we know that hemoglobin, which is a protein you find inside of our blood cells, it transports a tremendous amount of gas. And I've told you guys that, my experience with that earlier. But most oxygen, when it's absorbed in your bloodstream, binds to hemoglobin. Remember, hemoglobin's made of alpha and beta subunits, and that has to have a heme group. There's a central iron atom in the heme group, and that's the iron that can actually bind with oxygen and carbon dioxide. But oxygen will interact with iron on hemoglobin, and every hemoglobin molecule can transport 4 oxygen atoms, or molecules, right? Well, every red blood cell carries 250 million hemoglobin molecules. So that means that every red blood cell can carry up to 1 billion oxygen molecules, which is actually a tremendous amount of gas, right? Considering how many red blood cells you have per unit volume of blood. That's most of the oxygen is transported on hemoglobin. A very small percentage of it just dissolves directly into the water of your blood, the plasma. Okay, this is sort of the same concentration of oxygen that fish would rely on, right? You might wonder, well, how do fish breathe? Well, they're getting oxygen from their water. Well, they don't inhale air. They're actually just absorbing oxygen that's dissolved in the water that they are swimming around in. But it's a very small percentage. There's not hemoglobin in lake water, right? It's not keeping the oxygen levels high. But oxygen levels can vary from pond to pond, and what they're absorbing is the limited amount of oxygen that's dissolved in that water. In our bloodstream, it's only 1.5% of the total oxygen in our blood is the kind of oxygen that's dissolved in water, right? 98.5% of it, though, is bound to hemoglobin. So it just tells you how important hemoglobin is for oxygen transport. if you have any deficits of this protein, that can lead to some pretty deleterious effects. So what we're going to do next, you guys, is move on to talk about how oxygen is bound to hemoglobin and how that can vary under different conditions. Now, what this is showing is something called the oxygen-hemoglobin dissociation curve. Okay, so over here on the left, this is showing a molecule of hemoglobin, and each little red circle is supposed to represent an oxygen molecule. Okay, so we see that at its maximal level, hemoglobin can bind with 4 oxygen molecules. At its minimal level, 0. Right? Now, what we see here then is that this is an increasing concentration or pressure of oxygen, so that with increasing partial pressures of oxygen in blood, we see that hemoglobin becomes more and more saturated. Saturation refers to the degree of, of binding to oxygen. And it makes sense. If you increase the partial pressure of oxygen in blood, yeah, then hemoglobin is going to be more and more saturated. But you guys see how it's an S-curve here. Like, it's not linear relationship. So what this means is that under different degrees of partial pressure of oxygen, we get different degrees of saturation of hemoglobin that aren't predictable by a straight one-to-one relationship. It's actually it's a little different here. And what we see is that we see the largest differences in saturation in this kind of range here, guys, between 40 and 60 millimeters of mercury. So where was deoxygenated blood? What was its partial pressure? Yeah, about 40. And you can see here that has the steepest curve. It makes sense. It has the steepest slope, which means that with minor changes in partial pressure of oxygen, you get more dramatic changes in the saturation of hemoglobin. You might wonder why. What's the point of this? Well, for one, what this means is that as oxygen starts to bind, it makes it easier for more and more oxygen to bind. That's what we're seeing here. So you guys see that the slope here is not so dramatic. It's kind of a little bit more flat. But as oxygen starts binding and it becomes more and more saturated, then it's easier for hemoglobin to saturate more quickly. Okay. But what if I told you guys that this curve actually changes under different conditions? Like, this curve isn't always in the same position. You can actually get a right shift or a left shift, and it influences or changes how hemoglobin saturates under different degrees of partial pressure. And it makes sense with respect to tissues that might need more or less oxygen. So let's talk about this, you guys. So what this is showing is the oxygen-hemoglobin dissociation curve under different conditions. So this is the curve at 10 degrees C, 20 degrees C, 38, and 43 degrees C, right? So different temperatures of hemoglobin, and that can change this curve. Or down here, it's showing changes in pH. So in blue, this is if blood's at a pH of 7.6. In, I don't know, what is that kind of purple? This is if blood's at a pH of 7.4. And this one that's kind of hot pink is if blood's at pH of 7.2. What's the normal pH of blood?
7.4.
Yeah, about 7.35 to 7.45. So an average of that would be 7.4. Right? So this is showing the normal, right in the middle, right? So let's first talk about temperature. We know that temperature in our body's tissues can vary. Now, it's not going to vary from 10 to 43 C. There'll be more kind of smaller changes in temperature, but we do see that hemoglobin can change its properties based on different degrees of temperature. So why might a tissue be warmer than another tissue? What makes that tissue warmer? Yeah, it's more active. You got it. If a tissue is more metabolically active, it will produce more heat as a byproduct of its metabolism, which makes sense, right? You start breaking chemical bonds, heat gets released. So if it's a warmer tissue, that suggests that it's more metabolically active, so it's using more ATP. And if it's a cooler tissue, that suggests that it's less metabolically active, it's using less ATP. So just before I even jump into this graph, you guys, if you had to guess which tissue would need more oxygen, what would you say? The one that's more metabolically active or less metabolically active? More, exactly. So if you had a tissue that were more metabolically active, would you want hemoglobin to be more or less saturated as the blood moved through that tissue? Actually less. The reason I'll tell you guys why. If hemoglobin's saturated, that means hemoglobin's holding on to oxygen. If it's not saturated, that means hemoglobin's releasing that oxygen.
Okay.
So let's compare these graphs, you guys. So let's just pick a little reference point. Let's say 50 millimeters of mercury right here. So this is what hemoglobin will saturate at, different temperatures, but at 50 millimeters of mercury of oxygen, hemoglobin saturates differently, right? with different temperatures. So which one is the most saturated?
43 degrees.
No, that's the least saturated. Yeah. 40. This one's look at here. Yeah, look at the graph, you guys. So 40 at 43 degrees C and 50 millimeters of mercury, it's about 70% so saturated, right? How about 38 degrees C and 50 millimeters of mercury? Yeah, about 8, a little bit over 80, right? 85. Okay. How about 20 degrees C? Over 90, right? And 10 degrees C is off the chart. We don't even see that. So it's the least saturated when it's the warmest. That would make sense because if blood's going through a warm tissue, you'd want that hemoglobin to desaturate, to give up its oxygen into the tissue. And if blood's going through a cooler tissue, you'd want that hemoglobin to hold on to its oxygen and stay saturated. So what's really interesting about this process, you guys, you're seeing how hemoglobin can change its properties based on differences in temperature. And that's going to change how well it saturates. So if it's less saturated, where'd the oxygen go? In the tissue, right? Into the bloodstream, and it could diffuse into the tissue. If it's more saturated, where's the oxygen? It's still on the hemoglobin. So if it's still in the hemoglobin, is it in the tissue? No, exactly. So it makes sense with that regard. If like a warmer tissue is going to need more oxygen, and warmer temperatures have a tendency to desaturate your hemoglobin. Ah, interesting. Okay. So that kind of makes sense. And then let's look down here at pH. So if you look at the pH levels here, we see that we also have varying degrees. We get left shifts and right shifts. So earlier I mentioned how if carbon dioxide starts building up in water, it forms an acid, right? Carbonic acid. So if a tissue is acidic because of CO2 buildup, what might that say about that tissue? Is it very metabolically active or not so metabolically active? Yeah, it's more active, right? If a tissue is more metabolically active, it's going to make more carbon dioxide, which then gets turned into acid, carbonic acid, which makes the fluid in that tissue more acidic. So what if hemoglobin starts rushing through a part of your body that the tissue is more acidic than other tissues? What does that suggest about that tissue? Yeah, it's more metabolically active because there's more carbon dioxide building up, right? So let's compare these then, you guys. So we have 7.4, 7.2, 7.4, and 7.6. So the hot pink is 7.2 pH, purple here is 7.4, and then the blue line here is 7.6. Which one's more acidic? 7.2, yeah, exactly, which is a hot pink. So let's just make a reference point then. Let's say 60 millimeters of mercury. Let's just pick that as a standard reference point. So let's say at 60 millimeters of mercury of oxygen, let's go up. Which hemoglobin is the most saturated? The more basic one, right? And that would kind of make sense. If hemoglobin finds itself in a tissue that's more basic or less acidic, that suggests that the tissue isn't as metabolically active, so that it would make sense for hemoglobin to hold on to oxygen and not release it, right? Or what if hemoglobin found itself in a tissue that were more acidic?
It was active, so it would want to release it.
Yeah, that would make sense. You know, then you find yourself saying, well, if the tissue is more acidic, then it suggests it's more metabolically active, and you would want to desaturate that hemoglobin. That's what we're seeing here, right? So that at the same partial pressure of oxygen, hemoglobin's less saturated, which means it released its oxygen off hemoglobin into the tissue, which means that the cells can then use that. So the cool thing about this process, you guys, is that what it demonstrates is the fact that hemoglobin can selectively desaturate or selectively give oxygen to tissues that need it the most. It's kind of like supply and demand, where you have, well, actually, let's not use that example. It's probably better. Like it's actually probably better to say it's like socialism, right? You give more resources to people that need it the most, right? Like if people are starving, then you give them food. You don't give everyone the same amount of food, right? What if some people are starving and they're not getting enough food? Well, then you can give them more food, right? Let's think about that with respect to your tissues. Your tissues are highly metabolically active. They start getting warmer. Well, then hemoglobin gives oxygen to the warmer tissues because they need that oxygen. What if the tissue's cooler? It doesn't need as much oxygen. So at cooler temperatures, then hemoglobin's more saturated, right? Or what if the tissue is more acidic?
Less saturated.
That suggests that it's more metabolically active, which means that you want to release that oxygen in the tissue. Ah,